During this presentation, you will learn about qPCR.
I'm Kaylee.
I'm Maya.
I'm Jane.
And I'm Morgan.
So what is qPCR? qPCR, which stands for quantitative PCR, is also known as real-time PCR, and it is similar to conventional PCR in the sense that your goal is to amplify a particular sequence of DNA. However, qPCR is unlike conventional PCR in the sense that as the reaction occurs, the amount of product being amplified is measured as the reaction occurs in real time. After each cycle, the amount of product accumulated is quantified.
When setting up qPCR reactions, you combine the PCR reagents, custom primers, the reporter molecule, and the quencher. As DNA is being replicated, it is tracked with the fluorescent reporter molecule and is quantitative during this process. During this, the denature and annealing step are the same as normal PCR. However, it differs during the elongation. During elongation, the reporter molecule is close enough to the quencher to not fluoresce. When TAC moves along adding nucleotides, it will eventually knock off the reporter molecule and it will fluoresce because it is no longer close enough to the quencher. When the reporter molecule fluoresces, it is recorded by the optical module, and this measures the intensity of the fluorescence, and it converts it to a graph.
In real-time PCR, the accumulation of amplification product is measured as the reaction progresses in real time, with product quantification after each cycle. The qPCR workflow on the slide delineates the steps in real-time PCR. Reactions are then run in real-time PCR instruments, and the collected data is analyzed by proprietary instrument software, which allows you to determine the initial number of copies of template DNA or the amplification target sequence with accuracy and high sensitivity over a wide dynamic range. Real-time PCR results can either be qualitative, where there's the presence or absence of a sequence, or quantitative with the particular copy number.
When analyzing the amplification fluorescence graph that is created by the thermocycler, the curve shows the fluorescent signals as RFUs versus the cycle number, and thus shows the accumulation of amplified product. The curves can be broken down into 3 parts or stages. These stages of qPCR are the baseline phase, the exponential phase, and the plateau phase. The baseline phase is a part of the amplification process where little to no product has been produced. Therefore, the signal is below the threshold line. When conducting qPCR, there is a lot of background fluorescent signals during the amplification process. This background fluorescence is represented by the threshold cycle, also known as the CT value. The spot where your reaction curve intersects with your threshold line is the number of cycles it took to detect a real signal from your sample. So to explain this simply, the CT value is the quantitative point at which the thermocycler is detecting that there is more product being produced than background noise. Any real-time PCR run will have many of these curves from several samples, and therefore many CT values. The next phase is the exponential phase of amplification. This is where the reaction is doubling the amount of product that is being produced with every cycle. The final phase of amplification is the plateau phase. This is the part of the graph where there are no more reagents available to be turned into product. Therefore, the reaction has reached maximum production.
There are many different ways that qPCR can be used, but the most common one is gene expression analysis. Other uses involve identifying microorganisms, genotyping, diagnosing infectious diseases, genetic abnormalities, and cancer, determining bacterial or viral load, detecting numerous repeats within the human genome, as well as detecting and quantifying genetically modified organisms. qPCR is advantageous to use in the sense that it is quick and convenient, because the natural process eliminates the need to make a gel and perform electrophoresis. Since qPCR is a closed-tube system, there is less opportunities for contamination. The process is also highly sensitive and therefore open to error, and it's extremely expensive. qPCR is a method of amplification that gives you results simultaneously as the process is occurring.